My name is Chanel Jackson, and I will be presenting my abstract for the roles of protein disulfide isomerases in breast cancers. A little bit of background. Triple negative breast cancer describes a particularly aggressive type of breast cancer that affects African American women up to 3 times more than other ethnicities. Due to the higher rates of recurrence and poor prognosis, it is necessary to find a new therapeutic target for treating breast cancers. In order to conduct this research, over 40 research articles and review papers pertaining to PDIs and cancers were obtained from a multitude of published peer-reviewed journals. Each article was reviewed for insight on the roles that PDIs play in cancer cells, the design of the research conducted, as well as the diversity of the samples involved. Now, there are 4 aspects of note regarding this research. Number 1, PDI overexpression in each cancer cell line. Number 2, the roles PDIs have in cancer cell migration and invasion. 3, if PDI inhibitors are working on drug-resistant cell types. And 4, PDI inhibition sometimes improving survival rates. For this first diagram here, we can start with brain cancer because it meets all 4 of these 4 stipulations. For the glioma cell, PDI overexpression impacts the migration and invasion. Lower PDI expression is associated with higher overall survival rate in glioblastoma patients. PDI inhibition enhances TMZ-mediated cell death, TMZ being temozolomide. And PDI inhibitors reduce tumor growth of glioblastomas. And if we were to look at each of these other types of cancer cells, you can see here in this chart that there are similar roles and functions of PDIs in each of these cancers, such as colon cancer, ovarian cancer, lung cancer, and of course, bladder cancer. In this chart, we show the roles and functions of PDIs in various breast cancer cell lines. PDI members such as PDI-A1, PDI-A3, PDI-A6, and more are highly expressed in many of these breast cancer cell lines. As you can see in this chart, AGR2 and PDI-A3 show robust overexpression, and therefore treatments involving PDI inhibitors could suppress tumor growth for MDA-MB453. Over here for luminal A type, PDI inhibitors were reported to induce significant decreases in the adhesion and transendothelial migration of MCF7. For the T47D cells, high expression of PDI, ERP57, AGR3, and PDI-A6 were shown, but not ERP72. Excuse me. And for both of the basal-like types, which are also considered TNBC or triple negative breast cancer, PDI inhibitors showed adhesion inhibition in a dose-dependent fashion, even more effectively than PACMA31 alone. In conclusion, overexpression of PDIs is associated with breast cancer cell migration and invasion. Triple negative breast cancer is more common in premenopausal African American and Hispanic women. Cell lines widely used in breast cancer studies are mostly derived from Caucasian women, raising the issue of health disparities and their clinical implications. Thus, PDRs are promising targets for more effective diagnosis and treatment.